So here in Taylor's experiment, in the first step, as we see, we have a medium containing radioactive thymidine or thymine. Okay, we put the genetic information in this medium. Okay, after we washed, we observed on the autoradiography labeled DNA. Okay, we observed labeled DNA, labeled chromosomes. Both chromatids of each chromosomes are labeled. Okay, of course, this observation takes place during metaphase because chromosomes are visible, are the most visible during the metaphase. Okay, as we can see here, okay, the observation of cells at metaphase shows two chromatids, okay, of each chromosome radioactive. The thymine is a nitrogenous base that is part of the DNA molecule. When we label this thymine, we are able to observe the radioactivity in chromosomes, and we can see these radioactive red radioactive substances. Okay, fine. Of course, we observe these chromosomes during metaphase after allowing the cell to undergo mitosis, huh? After the cell have undergone the process of mitosis, we observe these chromosomes during metaphase, okay? Now, in the second step, we have washed the DNA, okay? We have washed the DNA. We didn't put radioactive thymidine. So what happened? Here we have the chromosomes. Half of them labeled, half of them not labeled. Why? Because during the second mitosis, DNA replication is semi-conservative. Not all the DNA is labeled. Half of them labeled, half of them not labeled because there is old strands and new strands. Okay? Finally, I want to explain briefly, because we explained this before, about the amount of DNA. I'm just gonna pause you 1 second. Again, briefly, this cell cycle. Here we have the interphase that is made up of three subphases, G1, S, and G2. Then after interphase, mitosis takes place. As we can see, the longest duration is for the interphase. During the G1 phase, chromosomes are uncoiled, okay, uncoiled. They don't have a shape, they don't have a good structure, okay? During the synthesis phase, chromosomes are being somehow coiled, and each chromosome start having two chromatids at the end of S phase. So we reach the G2 phase having a chromosome made up of two chromatids. After that, we have the mitosis. This is the prophase where the quantity of DNA is doubled. Now we have chromosome made up of two chromatids, so the quantity of DNA is doubled. During metaphase, the quantity of DNA is also doubled. This quantity is reduced from 2Q to Q during the anaphase until we reach the telophase, having the same amount of chromosomes but with one chromatid. After mitosis ends, we start again another interphase and cycle after cycle. Interphase, mitosis, okay? This is what I want to explain for today. But there is one thing I want to talk about here. What is the chemical constitution of a chromosome? When we talk about the chemical constitution of chromosome, we mean what is the chromosome made up of? The chromosome is made up of a DNA and a protein. The DNA and the protein. What protein we're talking about? Of course, we're talking about the histones that trap the DNA and make them in a coiled or condensed structure. Okay? Number one says the analysis of nitrogenous base composition of a DNA gave the following results: A22%, T24, C27, G28. Comment. What's your comment on this? I guess it's somehow visible that A and T, since they are complementary, their percentage is somehow very close. And C and G, since they are complementary, their percentage is also very close to each other, okay? I hope, guys, this video was helpful for you. I made this video to... I'm just checking something. Okay, I made this video to limit the wasted time in class in explaining these pages that I somehow explained on your copybook. So next time we will start with the new chapter. Good luck and see you. If you have any question, I will answer you on Thursday. Okay? See you.